Hello, I'm Peter from Stanford American International School, and today I'll describe a pipeline that predicts protein subcellular localization from amino acid sequence and then explains each prediction with something known as shapely additive explanations. Protein-based localization drives diseases from cancer to neurodegeneration, yet experimental assays are often slow and costly. In silico tools exist, but without clear rationale, their predictions lack impact in clinical and synthetic biology workflows. For instance, predictors like target P and D block achieve high accuracy, but operate as opaque AI black box models. No existing framework combines top tier localization performance with rigorous per protein explanation. Therefore, we ask whether we can number one, predict localization reliably, number two, explain every call in testable terms, and finally, number three, validate these explanations on held out data. Therefore, our solution pairs a k nearest neighbors KN baseline and a random forest RF classifier with shapely additive explanations, SHAP, to attribute feature contributions. To do so, we curated tens of thousands of UniProt knowledge-based entries across 16 compartments. Then we removed multi-localized and extreme length sequences, and finally performed a stratified 70%, 15%, 15% train validation test split to preserve real-world distributions. Our 422 features include amino acid compositions, dipeptide frequencies, gravy, grand average of hydropathy for core stability, and isoelectric point for charge-based targeting. This captures both local motifs and also global biophysical properties. So the KN baseline excelled at abundant classes but underperformed on rare ones. In contrast, the RF model, which consisted of 200 trees and a depth of 10, achieved uniformly high 1 versus red OX, which is around 0.93 to 0.97, and notably it boosted rare classes such as peroxisome from 0.74 to 0.86 and the vacuole from 0.77 to 0.85. Just to explain more about SHAP, it applies cooperative game theory to assign each feature a contribution value to a given prediction. By computing these SHAP values, we can obtain both a global feature ranking and also per protein force plots that can reveal why the model favored one apartment over another. So we have here that nuclear localization peaks at moderate gravities, which is approximately at zero, and this reflects a balanced hydrophobic core. You also have cell junction proteins predictions that correlate with high serine percentages. Um, Cytosolic assignments intensify with acidic isoelectric points, and that matches known enrichments of glutamate and aspartate. Finally, mean absolute SHAP values confirm that sequence length and isoelectric points do dominate model outputs, while select dipeptides such as AE and 2N do contribute to finer compartment distinctions. Let's look at cytosolic protein signatures more specifically. So SHAP actually reviews three cytosolic charge subgroups, acidic, neutral, and basic, and each reflects adaptive roles. You have the acidic residues that enhance solubility, and moderate hydrophobic cores ensures folding without membrane insertions. Proteins that are exceptionally long are penalized, which cooperates with the fact that cytosolic enzymes and domains seldom exceed moderate sizes, in keeping with processing and import constraints. Altogether, these attributions recapitulate well-established biochemistry, and it demonstrates that our explainable pipeline not only localizes proteins, but accurately does so by learning genuine molecular principles. Clinically, our pipeline can flag variants that disrupts localization signals and integrate these SHA profiles into ClinVar for transparent pathogenicity assessments. However, limitations do include sparse data for rare compartments, and there is this need for models that capture long-range sequence motifs. Therefore, we will implement a one-dimensional convolutional neural network to learn discontinuous motifs, deploy a web dashboard for interactive protein exploration, and extend our framework to metagenomic and non-model organism datasets. So that's all. The code is open source at Cytronic/Explainable Localization Predictor, and we welcome any contributions to the work. Thank you so much. Have a good day.